Hello again, guys. Happy Monday. I am recording on Monday. I know I told you to look at this recording on Wednesday, but again, since I'm recording today, happy Monday. First thing, I just want to go over everything just to get some clarity. Remember, all of our teachers are on the same page. We should be available between 11:45 and 2:45 with the Google Meet. All you have to do is hit me up. Just send me an email. Let me know. I'm not just going to pop on Google Meet. But if you scroll on my main page, remember everybody has their own Google Meet link per class. So I'm just in second period just because that was my first class that I popped up. But all you need to do, send me an email, say, hey, Miss Swann, meet me on Google Meet, or can you jump on Google Meet? Click on your link, and then I'll jump on and talk to you. I know you guys miss me so much, like you miss all of your teachers. Anyway, if you go to your bulletins, of course, you click on the image. It will pop up the matrix for this week. Of course, everybody's been staying on top of it. Thank you. If not, I've been emailing you guys, keeping up with everybody. You already know we're in the same routine. I don't really want to switch up and add anything that I haven't done since you met me in August. So this week, what we have going on again today, which again, I'm recording on Monday. So Monday, you had to go sign into your textbook. There were two 6.3 assignments. One of them you could redo, and one of them you couldn't. Remember, anytime you click on those, the one that says teacher, you can redo. The one that says quiz, you cannot redo. So just make sure that you're understanding that. Same as normal. Those are our normal warm-ups. It's just the 5 questions. After that, I asked you to read section 6.4 and complete those PowerNotes and turn them in. You also have another assignment for Tuesday that, again, is in the textbook. It's 2 of the assignments for section 6.4. Then what I asked you to do is get the reinforcement worksheet done for section 6.4. Please read those questions carefully. I know some of you guys with the last section got number 1 wrong because it asked you which generation, and you were telling me a 3.1 ratio. Something about it. That wasn't the answer to the question. So just make sure you're paying attention to that. And then, of course, I'm asking you to complete the concept map. Today, which is Wednesday for you guys, as you are listening to this video, I am asking you to go through, make sure that everything is up to date on your 6.4 PowerNotes. So what you're going to do is you are going to go to your PowerNotes. Remember, I went through page 1 for everybody. That was the mitosis versus meiosis. I went through 6.2, which is how we did our group quizzes. Those of you that did not do the group quiz, I sent you an email. Don't forget, click on that link, log in, take that quiz for me. I'll see the update as soon as you do it, so you don't have to let me know that you did it. Remember, this is where we did our group quiz. We took our pipe cleaners, our pop beads. We did all of that stuff and made those. I put those grades in. If you need to see those, I'll email them back to you. I'm pretty sure that I did that for everybody. If you didn't, let me know. Last week, I went over section 3. We met Mr. Mendel. Gregor Mendel is the father of genetics. Remember, he studied pea plants and genetics amongst those pea plants. What he's doing is he's coming up with these crosses. These crosses are put into like little math equations known as a Punnett square or a monohybrid cross. We've got a parent generation, an F1 generation, F2 generation. The conclusion, basically, he found out that our traits are inherited in our genes. We have some that are discrete and some that are expressed. Remember, he mated the 2 or crossed the 2 purple flowers, ended up with all purple, crossed those, and then ended up with some white. Basically saying that our purple flowers are going to have a hidden white trait. For today's notes, our 6.4, we're going over genes and alleles and some of our basic vocab. First thing, genes, remember those are the pieces of DNA that tell the cell a certain polypeptide to make. And that polypeptide, remember, has to do with our DNA. Our DNA is found on our chromosomes. Remember, this here is a chromatid. So all of those little sections are DNA. Here is a picture of the actual karyotypes. Remember, we took those sections, matched them up. So each segment, all those bands, those stripes are the actual genes themselves. So again, definition of a gene, it's that piece of DNA that's going to tell the cell to make a certain polypeptide. That polypeptide is therefore going to find its spot on the chromatid and so on. We'll talk about the details of DNA later. Our alleles. Alleles are different forms of a gene. So from here on out, we're going to write our alleles as letters. You need to make sure that they're going to be different forms. So therefore, we're going to use the same letter when we're talking about the same gene. I'm going to scroll over here really quick just to show you. For example, T for tall, capital T for tall. Let me say that correctly. Lowercase t for short. We're not using an S for short because T is obviously our dominant trait. It's also the gene of height. So because we're talking about height, we have to use the same letter. So we have different heights. We have tall and short. Which one is dominant? Tall is dominant. So we're going to use T. Capital T for tall, lowercase t for short. Again, we cannot use a capital T and a capital S or a lowercase s because that would mean we're talking about two different genes. In this case, we're only talking about height. Same thing with yellow. Yellow is dominant over green when we're talking about plants. Peas, for example. Remember, we're talking about green peas and yellow peas. So yellow is dominant over green when we're talking about those plants. So we're going to use a capital Y and a lowercase y because that's the gene of color. Anytime you're talking about the same gene, you have to use the same letter. Capital is going to represent dominant. Lowercase is going to be recessive. Kind of getting ahead of myself, but talking in the same box of alleles. So there may be many different forms of the same gene in a population, but we always use the same letter if we're talking about the same trait. Each individual is basically going to have those two forms. You get one from mom and one from dad. So this is how, again, guys, if you end up becoming bald in the future, you can thank mom. And that's if dad is not bald. Why? Because mom is going to carry that trait on her X chromosome. And what she's going to do is she's going to pass that trait to you because you only have one X. Remember, guys, you have an XY. Ladies, we have two X's. So we have more of an advantage when it comes to certain traits. Ladies can be carriers of traits on the sex chromosomes and guys cannot. So again, you get half of your traits from mom and half of your traits from dad. This is how every individual has two alleles. Those two alleles, again, represent the one that you're given from mom that's dominant or recessive and the one that you're given from dad, which is dominant or recessive. Anytime we're talking about our genes, if you notice, I carried everything over here on the left in the green. So genes all laid out together make up what's called a genome. We have the Human Genome Project, which is this idea of mapping out all of the human genes to try to make the ideal human being. We want a person that lives forever, doesn't get sick, dies of natural causes, and we don't have to worry about coming up with vaccines or cures. Of course, look at where we are now. What a coincidence. Talking about the coronavirus, trying to find a cure, going through, again, genetic testing, age right now, the ages that it's affecting, and so on. So a genome is all of the organism's genetic material. So all of the possibilities. Going on this side, if we pop this up, I'm just going to add in here and I'm going to type in the Human Genome Project. That is going to take us to the actual Human Genome website, which is here, genome.gov. And there's a lot of information, the research on the human genome. So it gives you all of the information, basically what is it, what are the results. So it says so far we have a sequence of 3 billion DNA letters in the human genome. Blah, blah, blah. It's going to give you even more, the achievements and so on. You can play around with this website and look at even more. Again, on your own time, the Human Genome Project is at genome.gov. I'm going to switch back over to cut my time with The word gene, we're going to use that prefix Gen, Geno. So we have a gene, which is a segment of DNA, a genome, which is all the genetic material laid out. Again, those 3 billion segments of DNA or DNA letters, the A, T, C, and G that we talked about before: adenine, thymine, cytosine, and guanine. And then we have genotype. Geno, again referring to gene, it's the genetic makeup of a specific set of genes. Anytime we're talking about a genotype, we have 3 different collaborations or ratios or vocab words or ways to write out a genotype. You have your homozygous dominant, which is going to be your 2 uppercase letters. Homo, remember, means same. Zygote, referring to the fusion of those chromosomes. So same chromosomes, same genes. Heterozygous, it's going to be different. Hetero is other, different. So 1 uppercase, 1 lowercase letter. Has to be that option and that option only. Homozygous recessive is going to be our 2 lowercase letters. Homozygous, again, meaning same. Recessive, meaning lowercase. So again, we have dominant, which are uppercase. Recessive are going to be lowercase. When we're looking at the differences in those definitions, homozygous also means pure, like a purebred animal. That describes those 2 alleles at the same chromosome location that are the same, whether they are 2 uppercase or 2 lowercase, as long as they are pure. Homozygous, again, 2 uppercase or 2 lowercase. I gave you some examples on the side. 2 uppercase T's, if we're just using T's as an example. Same case, both upper. Again, homozygous, the same. 2 lowercase, same case, both lower. Homozygous, no matter what, homo means same. You either have 2 upper or 2 lower. On the flip side, our heterozygous is also known as our hybrid. This will be like our mutts when we're talking about dogs or any type of animal that's mixed is going to be a hybrid. Heterozygous, hetero means other. It's going to be those 2 alleles at the same location that are different. So only option we have is a uppercase letter, lowercase letter, always written in that form. So for example, with your name, I'm just going to write my name over here on the side. I always write my name like that. I would never write my name like that. That is a big no. Why? Because capital letter always comes first. So anytime you are writing alleles, the uppercase comes first. Just remember, the way you write your name, you do not write the uppercase letter second or third or last, because that's weird and we don't do that. Moving on to the right side, because again, I just went, all the gene stuff is in green. So gene, the piece of DNA, I went over and down. Genome, genotype, 2 types, again, homozygous or heterozygous. And then I'm going to come back up to the top because the phenotype goes for both. So I'm going to come back up here to allele. Our allele information is going to be in the blue. So remember, allele is a different form of the gene. Over and down, they are represented by letters. We're going to use uppercase letters for those that are dominant and lowercase letters for those that are recessive. When we get to our next assignment, which I'm going to show you before I end this video, it's basically going over vocab words. You have to know heterozygous, homozygous. You need to know dominant. You need to know recessive. You need to know allele. You need to know phenotype, genotype, and hybrid and pure. Those are the words that you're going to need to know from here on out. Anytime we're working with monohybrid crosses, then we'll get to dihybrid crosses, which are kind of difficult. Hopefully, I'll be able to Google Meet you guys to do that so that you can see me face to face and we can actually talk it out, because it's pretty difficult when we get to the dihybrid. So anyway, getting back to alleles. Alleles, again, are represented by letters. Allele, letter. Just remember that. Uppercase or lowercase. Dominant alleles are the ones that are expressed, the traits that you actually see. So the physical traits that you have, if you have brown hair, you have blue eyes, you have hazel eyes, you have green eyes, you have blonde hair, brown hair, red hair, ginger hair, All of those different options, the ones that you actually have are the dominant traits. The recessive traits are the ones that we're actually only going to see if both recessives are present. Your examples, anything that's dominant is going to have a capital letter in it. You have two options. Your homozygous dominant, which is going to be your big T, if we're talking about tall. But also heterozygous is going to be tall because of that capital T. Big T, little t. Anytime there's a capital letter, it's going to represent a dominant trait. The recessive, again, only has one option. It's just our lowercase letters. Little t is short. Why are we using T's for short? Because remember, we're using height. Height is the gene. The dominant gene of height is tall, so we use a T. So you have two options, tall and short, big T, little t. That's why we're doing that. Bring it all together. It's going to work us through what's called a phenotype. Pheno, think physical. Pheno and physical. Phenotypes, again, represent the physical characteristics that we see through either the genotype or the environmental factors. So whether it's tall, short, green, yellow, wrinkled, smooth, round, those are talking about the pea plants that we looked at with Mendel. All of those things are going to be represented in the activity that you have to do. Let me see, because I don't remember if it is tomorrow, meaning Thursday, or Friday. Let me see what we got. Bikini bottom. Bikini bottom genetics is what you have to do on Friday. So what I'm going to do, of course, Thursday, tomorrow for you guys, you're going to have to do your Amoeba Sisters. That's just a recap. You can do the worksheet if you want a little bit of extra credit. It's not going to be worth a lot, but for some of you, it might help at the end of the quarter, which should be done on Friday, but we'll see what happens then. So anyway, what I'm going to do is I'm going to walk you through the Bikini Bottoms worksheet intro. Remember, you have to make me a video just like this. You're trying to figure out what to do. If you swipe down from the corner, this little button that's blinking, if you touch and hold it, it'll pop up a little menu, and I don't want to do it because it's going to stop my video. And all you have to do is click microphone on, and then you can start screen recording. And what I want you to do when you're doing that is you're going to go to the resources. Resources over here. No matter what your class is, under chapter 6, we have, where did it go? It's not up there. Let me add it for you. Bikini bottom genetics. We're going to put that again in our chapter 6 folder. And we got to find it. See what we got going on. Bikini, go back. All right, timeout. I'm going to get it from here. I'm going to have to upload it for you guys. Mendel and Punnets. Bikini bottom genetics. All right, so again, I'll upload that. I'll put that there so you guys can have this in the resources. It'll already be available. But anyway, what you're going to do is you are going to start a screen record for me. If you read your directions, it tells you basically we're looking at the genetic makeup of organisms in this community. Everybody knows SpongeBob and Patrick and Squidward and Mr. Krabs. But put your name on your paper. And then you are going to tell me, okay, for each genotype in number 1, you're going to tell me whether it's heterozygous, H-E, or homozygous, H-O. We're going to be mature about that. Sorry, it says ho. But I want you to make sure that you are understanding this here. 2 uppercase. Are they the same or are they different? Those are the same, so we're going to call them homozygous. this here. Are those the same? Two uppercase or two lowercase? No, they're not. So they are heterozygous. You're going to do this for every single one. This is an uppercase and a lowercase. Therefore, it is not the same case. So we're going to call it hetero. Hetero meaning different. Lowercase, lowercase. That is the same. There you go. Keep going through those. You've got a couple at the top. Which of the genotypes would be purebred? Purebred is the same as homozygous. Pure and same. Remember, that was from our notes. If you're trying to figure out where. Here. Homozygous, pure. Right there. And then heterozygous, hybrid. So, going back to this, which of the genotypes? And number one will be a hybrid. I'll give you a different color. Hybrid is heterozygous. So you're going to tell me which letters. So basically all you're going to do is purebred is going to be our big T, big T. It'll be our lowercase f, blah, 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 blah. And then you'll do the same thing for your hybrids. So number two, determine the phenotype physically. Yellow is dominant to blue. This big Y, yellow obviously represents the dominant. So this would be yellow because there is a capital Y present. What about this one here? Because there is a capital, it has to have a dominant trait. So what you're going to see is you're going to see the yellow, but it's going to carry the blue. So that's what that means. It carries that. But it does not show it. It only carries it. And then the last one, little, is going to be blue because that's recessive. So you're going to keep going through number two. For number three, walking you through what tall is going to represent. It tells you there. Tall is going to be a capital T. Short is going to be a lowercase t. Why? Because we're talking about the gene of height. So it has to be the same. Pink body for Patrick is going to be capital P. Yellow body is going to be a lowercase p. Please make sure you designate the differences in your capital and lowercase, because now what we have to do is in number 4, we have to figure out, and again, this is your homework, so you should be doing, or sorry, not your homework, your classwork for Friday. So you're going to be making me a video just like this. SpongeBob recently met Sponge Susie. She's round, he's square. It says there, SpongeBob is heterozygous for his shape. Hetero. He's going to be a square. Heterozygous. Big S, little s. Why did I say S? Because he is a square pants. Why did I say big and little? Because he is heterozygous. And why did I put it on this side of the Punnett square? Because that's how Punnett squares work, like multiplication tables. But Sponge Susie is round. So our big S is going to be square. And our little s is going to be round. She has to have 2 alleles, one from mom and one from dad. Because she's round, they both have to be lowercase s's. And then we use this like a multiplication table. These go down and these go over. So we're going to have big S, little s. Why? Because I don't write my name lowercase a, capital N, G-I-E. No, I put the capital first. And then both of these come from the top and the side. So all possible genotypes, the genotypes are going to be the children. The children are what's found in the inside and the inside only. Because again, a Punnett square shows the probability of children. So possible genotypes, big S and little s is 1 because you get one from mom and one from dad. They got the big S from SpongeBob and the little s from Sponge Susie. And then the other possible genotype is going to be little s, which we get one from SpongeBob and one from Sponge Susie. Possible phenotypes, we're going to be square and round. Now, what are the chances that you're going to have a child with a square shape. We're only looking at the yellow. Because remember, SpongeBob and Sponge Susie are the parental generation on the outside of the Punnett square. So the chances of a child with square shape is going to be this here and this here. And I'm just going to do that. So what was that? That's 2 out of 4. How many boxes are there? There are 4. 1, 2, 3, 4. And then the chances that we have a child that's round, the other 2. 2 out of 4. 2 out of 4 is 50%. 2 out of 4 is 50%. And that is it. So I'm going to walk you through number 5, and then you can continue. Patrick and Patty met at a dance. They are both hetero for pink. We already know that pink is dominant. Why? Because it said it up here. Pink body. So what letters are we using? P's. P for pink. If they are both heterozygous, that means we have to use 2 different cases. So therefore, they are both big P and little p. And I'm just going to make my little key over here. So big P stands for pink. What does little p stand for? It is their yellow body. So little p is going to be yellow. Again, it's kind of hard to think of that because when you see a P, you want to think of something that begins with a P, but it's not. Because this is the gene of color, we have to use the same letter. If you have any questions on this, again, on Friday, you can Google Meet Me. But as of right now, that is everything you need to know for 6-4. I'm flipping back over here to make sure I covered everything. Did I get off of what I needed? Yes, I did, but I'm going to open it back up. So this is Wednesday's recording. This is Miss Swann 6.4 PowerNotes lecture and some, because again, I just gave you that information on how to get your Bikini Bottoms worksheet done. It says create an audio screen recording. So if you need me, hit me up on Google Meet. Again, you guys relax. Don't stress. Take your time. Let me know how things are going. Check in with me every once in a while. Some of you I heard from like every day. Thanks for the emails and the little comments in your portal. The rest of you, it's cool. Get your work done. You don't have to talk to Miss Swann as long as I see your work being uploaded into the portal. Until then, I will talk to you guys later. Happy Coronication. Stay safe. Wash your hands. Lysol and Clorox wipe your area like we do in the class. Talk to you later. Bye, guys.